Here I'd like to welcome Eva Loth for our next topic is experience of an autism researcher, how concepts, trends, and autistic people have influenced my work.
Thank you.
Welcome, Dr. Eva Loth. I have to introduce Dr. Eva Loth is a professor of cognitive neuroscience at the Institute of Psychiatry, Psychology and Neuroscience, King's College London. She received her PhD in psychology from St. Andrews University, Scotland, and completed postdoctoral placement at the IOPPN and Cambridge University. Her work combines developmental, cognitive, and neuroimaging approaches to better understanding the relationships between social, emotional, motivational, and cognitive processes in typical development and neurodevelopmental conditions, such as autism. Eva is also the Deputy Director of the AIMS 2 Trials Consortium, which aims to develop precision medicine for autism. So over to you, Eva.
Thank you very much for the kind introduction, and thank you very much for the invitation. I so enjoyed this conference and the presentations this morning and yesterday. Let me see if I can share my slide. I hope that's the right presentation. Okay, can you see my screen? Great, okay. So I would like to thank in particular Dr. Mary Doherty, who invited me to speak at this conference. And Mary asked me to talk about how the neurodiversity paradigm and neurodiversity movement had influenced me as a researcher and my research. And when I was preparing for this talk, I realized that many of you may not be familiar with my research. And so for this reason, I thought I am going to divide my talk into 3 parts. I will first talk about my education and early career. Then I'll talk about the last 10 years or so that I was working on AIMS to trials, or EU AIMS and AIMS to trials. And then where I'm now and in the future, I'm trying to integrate neurodiversity affirmative approaches with precision support. So when reflecting about, my journey as an autism researcher, one thing that really stands out is how much has changed in what we thought we know about autism over, in my case, these last 20 or 25 years. And that includes even the fundamental question of what is autism, who are we studying, and also how we conduct our research. And I'll start by telling you a little bit about my background. I didn't actually start off as a researcher in biomedical science, but I started off as a student in sociology, and I was broadly interested in sort of social behavior. I was first in the south of France, and then my parents felt it might be better for me to maybe come back to Germany, where I was from. And I went to the University of Trier, and I switched to psychology, but carried on with sociology as well. And I wanted to relate to you two experiences from that time. The first one was when we went to our induction week, as it's called, our very first lecture. And the first professor came up and said, well, as you know, psychology is fundamentally a social science. And then he proceeded to tell us all about what he was going to lecture. And then we all agreed. And then the next person came up, the next professor, and he said, well, as you all know, psychology is fundamentally a biological science. And then he proceeded to tell us what he was going to lecture us about. And there was no attempt made to reconcile these different views or to try and integrate why one approach might be helpful for the other. And I think this is also still relevant to how we look at autism. But I then moved on after 2 years to St. Andrews University in Scotland. The second thing I was going to tell you about is a lecture we had about the sociologist Pierre Bourdieu. And he'd carried out in the 60s or 70s a series of studies where he asked people from different walks of life what they saw in different paintings and different pictures. So here, for example, the picture of these praying hands. And one person might have said, they may have reflected on the hardship of old age. Another person may have commented that they might found these hands ugly. Another person may have noted the intricacies of the drawing. And Pierre Bourdieu used these studies to make the point that taste, which is something that we often think of as being so individual, is actually to some extent socially determined and helps to perpetuate social inequalities by virtue of having a particular way, a mode of perception or thought, how one sees things and interprets experiences. And I guess what stayed for me more than that even is more generally how different people can perceive and interpret the same things differently, and that different things might be relevant or meaningful for one person and another person. I then moved on to St. Andrews University, initially with the idea to stay there for 1 year, and I have been in the UK ever since. And there I learned for the first time about theory of mind. And we've just heard in the previous presentation all about theory of mind and also the dangers that the account had brought along with it. But back then, in 1996, it was a very dominant approach in developmental psychology, where people were asking the question of how do children learn to understand different people's mental perspectives. And of course, there was also the very influential, very dominant theory that autism involves theory of mind deficits. Now, at this point, I had not met consciously an autistic person, which was not surprising because at the time we thought that maybe 3 or 4 people in 10,000 people were autistic. And I was very fascinated by this account, but I was also interested in autism because it seemed to involve not only difficulties, but also very particular strengths and talents. So after I graduated as an undergraduate, I went to London to work with Francesca Happé, but also to work in an autism school or a school for children with special needs. And I was almost expecting from what I'd read about autism to meet children who had theory of mind deficits. But of course, the reality was a little bit different. I saw a lot more diversity in those children. And I also realized I couldn't even test many of these children on these traditional theory of mind tasks because many of the children were perhaps not able to, sit through the story or understand the story or didn't find it interesting. On the other hand, I started helping out in a support group then back in Scotland for teenagers with Asperger's syndrome at the time. And of course, many of those could understand these very standard theory of mind tests. So I then embarked on my PhD, and we were trying to look at theory of mind not only as a capacity, but also, what impacts how different people use theory of mind. And on the one hand, we were interested in how autistic people would experience different kind of social events with and without theory of mind. And again, as we've just heard, there was another theory that was very influential, which was called weak central coherence, the idea that autistic people perhaps are more focused on specific details, perhaps at the expense of integrating information in context, and how this might impact on how events are experienced, everyday events, but also how one then uses a knowledge of events, so schematic representations, in order to, selectively draw attention to different things or to interpret experiences. And at the time, really, a lot of my study participants were the children and the young people that I saw in the support groups, and they were, in a way, my reality check of what we were studying may or may not have made sense for them in their everyday life. But then the second part of my PhD went back to an interest in sort of intergroup relations. And in particular, we started off, being interested in what made the Holocaust happen. And I worked with Steve Reicher, a social psychologist, and one of the very influential theories for a long time was that maybe it had something to do with the characteristics of the German people at the time. But the tradition in which Steve has worked was in something called social identity theory, which was coined by Henri Tushwell and John Turner. And they proposed that one can define oneself at different levels of abstraction, as I versus you, as we versus them, perhaps as a mother, perhaps as a professional, and which part of one's social identity is more salient would depend on the context. So we were interested in the question of whether how we use our theory of mind would depend on the relationship between oneself and another. And we were predicting that if one is in a more powerful position, one can afford to be less empathic and also to lack more self-reflexivity with regards to the impact of own actions on others. Whereas if one is in a powerless position where one's own fate is controlled by the powerful, one has to understand the mental perspective of the others in order to basically survive. So it's not completely lost on me. At the time, I didn't really integrate these two parts of my PhD. I did write a concluding chapter, but now it's not completely lost on me that some of these, observations and interest in intergroup relations also are applicable to the way, the scientific community, the medical community, and autistic people relate to one another. But this was something that I hadn't been able to predict at the time. But it echoes to me a lot of what we now know as part of the double empathy theory. So in summary of this part, so a lot of it was sort of the research was driven by theories, and it was embedded in particular assumptions that different kind of theoretical approaches were making, including in terms of autism, also the very categorical view on autism, which sort of divided autism quite strictly from ADHD and other neurodevelopmental conditions. The work on the whole was relatively small scale, and in my case of low impact or no impact, because I'd never published a work on theory of mind and intergroup relations. But at the time, also, I was interested in better understanding what might underpin differences in cognitive development. I was originally hoping to be able to further pursue the work on autism that I then extended with Francesca Happe, but I wasn't successful in getting another fellowship, so I was looking for another job. And I was interested and intrigued by the emergence of neuroimaging methods that had come out at the time in order to study the living brain. So basically, I then trained in neuroimaging at Cambridge University in the context of a social affective neuroscience group, particularly in how the brain processes rapid changes in emotion processing, and then went on to work in a large-scale gene neuroimaging study at the Institute of Psychiatry. That followed about 2,000 adolescents from the age of 14 years to look at early predictive markers of a range of psychiatric conditions with onset in adolescence, including addiction, anxiety, depression, externalizing, internalizing difficulties, and so forth. So I kind of went out of autism research for about 5 or 6 years. And when I came back to that, a lot of has changed. I had very briefly, during the time that I was working with Francesca, also met Professor Declan Murphy, but I didn't think I made a big impression on him, and I was rather surprised that he invited me to join the EU AIMS project. And he had just received this grant as the primary principal investigator, which was at the time the largest project focused on precision medicine for autism in the world. So the landscape had changed quite a lot since I had worked on autism before. On the one hand, there was now an increased prevalence of autism that was recognized. So was the diversity among autistic people. So when beforehand, the main question was, what do autistic people have in common and what is different to other neurodevelopmental or neuropsychiatric conditions? Now the question had moved to, what can help us to explain the diversity among autistic people? And also it was more recognized that autism involves also co-occurring mental health and medical conditions. Now, EU AIMS was very different for not only myself, but many of my academic colleagues, including many more senior colleagues, in that it was a public-private partnership, part funded by the European Commission, but also partly funded by industry within the umbrella of the Innovative Medicines Initiative. And the interest from industry in that stemmed from the recognition that many drug trials had failed and that one needed to work together in order to make more progress, in order to understand the basic science, neuroscience. It is maybe worthwhile mentioning that the Innovative Medicines Initiative only funds biomedical research. So in a way, the argument that has been made, why wouldn't these funds be used for something that might be of a higher priority to autistic people, was in some ways not a matter of debate, because they just wouldn't have funded research on more social aspects. It was a matter of either autism or perhaps they would have funded another completely different medical condition, as they have predominantly done before, like cancer research or infectious diseases or others. So the notion or the motivation for precision medicine was really something that a concept that had been imported to psychiatry in general, and then autism from internal medicine, so oncology and other disciplines within internal medicine. And really, because across the board, it is the diversity, the heterogeneity that has been a stumbling block in successful treatments or interventions. In the case of autism, it was recognized that a diagnosis of autism alone doesn't enable us or professionals to make very accurate predictions about, for example, a person's development or developmental outcome, whether one person who is non-speaking in their early childhood would also carry on to have a lot of support needs as an adult, whereas another person who might present very similarly, perhaps over the preschool ages, might then go on to, become a very highly successful professional, etc. Also, an autism diagnosis alone doesn't help us really to make good predictions of what might be the treatment needs, if any, of a particular person, what might be the benefits of a particular approach, and also in the case of medical treatments, what might be the side effects. And it's well known that in autism, medications are used for other conditions may sometimes have different or stronger side effects. And also, the autism diagnosis alone doesn't tell us much about what might have caused the condition in this particular person. So the goal of precision medicine is to identify new mechanism-based treatment targets and then to tailor those treatment or interventions to individuals using so-called biomarkers. So what are biomarkers? So a biomarker is a little bit of an elusive concept, and it could be in principle any test or measure that helps a professional to make clinically relevant predictions about a person based on some biological characteristics. And it could be really anything from a cognitive test, a brain scan, something that is measured with eye tracking, the microbiome, immune markers, as well as genes. And people often differentiate different types of biomarkers by virtue of how there are, what is their likely or intended clinical application. And that ranged from, likelihood markers to predict the likelihood of an infant to develop autism, perhaps before behavioral features emerge. It could help in the diagnosis, a more objective or reliable diagnosis, help in making a better prognosis about the progression. So, for example, whether somebody might also develop co-occurring ADHD or anxiety, and to predict which therapy might be beneficial for a particular person. Now, we know that autism is a diverse or heterogeneous condition. So any of these kind of types of biomarkers that I've just mentioned are likely only applicable for a particular subgroup of autistic people. And they may or may not be overlapping with people who have another primary diagnosis in the case of prognostic or predictive markers. So in order to identify or discover biomarkers, we created the Longitudinal European Autism Project, or LEAP. That was somewhat different to many of the research studies that were done before by virtue of the scale. So in total, we included over 700 participants, more than 400 autistic participants from children, adolescents, and adults, and as well as a comparison group that comprised both individuals with intellectual disability without autism and those with so-called typical development. And each person was characterized quite comprehensively in terms of their clinical profile, including also co-occurring conditions, quality of life, cognitive profiles using a set of, social and non-social tests. Some of them used eye tracking, brain structure and function, biochemistry, and then later on, we also had received additional funding for genomics. And it's a longitudinal study in that each volunteer, if they were willing to come back, were followed up now for 3 times. So the third wave is ongoing. And we're hoping to finish that at the end of this year. The other thing that was different for us was that we really intended to use these biomarkers in clinical trials or in clinical practice. Now, for many academics, the end goal is to publish a paper. But if you wanted to use biomarkers that were used in the clinic, then they had to meet several criteria that are somewhat more stringent, so that they would have to be validated, they would have to be replicated, it would have to be robust, so that they could be used. And for this reason, we engaged with regulators to understand what were their requirements for the use of these biomarkers. Now, the first kind of set of analysis we done were quite traditional case control differences, where you have one group, in this particular case, the autistic group, and a comparison group, and you look at what are the mean group differences. And one thing that we noticed was that in the whole of the autism literature and in many other conditions as well, people often misinterpret mean group differences. So basically what you usually get is an average group difference. And then it would be that autistic people would have on average, for example, larger brain structures or much more or less functional connectivity or theory of mind deficits or whatever it is. But then people often forget about the on average and then say, autistic people have whatever it is, larger brain volumes or connectivity or so, which has a very different meaning. And they might even be compelled to then, say something like autism is characterized by this particular feature, or that it would be even a biomarker for autism. And what we've shown with some simulations is that we really need very large effect sizes in order to have something that is truly diagnostic for a condition. But when we looked at our own results from this large study and also compared that to meta-analyses from previous studies, we showed that the effect sizes were such that there was a very large overlap between the autistic and the non-autistic group on pretty much any measure that we've used, which actually translated in the finding that it was roughly between half of the autistic people or over half of the autistic people who would fall right within the normal range of the comparison group. So there would be only a small percent, or not a small, a percentage of autistic people who have any of the postulated differences or deficits. So there was no characteristic that would meet the first criterion for a diagnostic biomarker. And so that means that we really need to find better ways to make predictions about an individual. And interestingly, the same observation also pertains to something like quality of life, which one of my previous PhD students, now a lecturer, Beth Oakley, had been interested in, and she looked at quality of life in that cohort in children, adolescents, and adults. And although there were mean group differences, that doesn't mean that all autistic participants, either self-reported or reported by their parents, had reduced quality of life. But when this was the case, the strongest predictor was anxiety, followed by depression. So a lot of work by my colleagues went on in creating new methods in order to make better predictions about a person, and that included to have scores that are comparable for different people that are normed in that way, in order to be able to situate one person in terms of expectations relative to their age, relative to their sex or other characteristics, and then use more what we call data-driven approaches to see whether we could identify some subgroups. And this is exemplified here with the example of the N170. This is a brain signature of how the brain responds to faces. And we found that this particular N170 latency was slower, again, only in a subgroup of autistic people, which was then also related to another measure of brain function using fMRI in a particular brain region, as well as polygenic scores, as well as this lower response to faces in this subgroup of people seemed to help to predict social prognosis over a period of 18 months. And so what we included was cautiously, it's not at all a biomarker for autism, but it would help to understand who might be naturally making gains or find it harder over time. And those might then be the people who might be more, for whom it might be more relevant to have some support. Okay, so the main achievements from this part was that this multidisciplinary approach allowed us to look at one person quite comprehensively across different levels to increase robustness and reliability, which is important in what is sometimes called a crisis in neuroscience. We engaged with regulators, dispelled some myth, and created an infrastructure for European clinical trials. But we did not work very closely with autistic people, and we were to some extent aware of some of the criticisms And the question was whether some of these goals could be aligned with priorities from the autistic community. So we then were able to get another grant, an even larger grant, to carry on and to expand this work program. But we wanted to make a number of changes based on the experience with EUAIMS. The first one was to include autism representatives in the project. And we also moved from case control studies to cross-condition designs. We shifted from the focus on core features to greater emphasis on mental health, sleep, sensory processing, and epilepsy. And this was partly also informed by a survey by Autistica, looked at what are actually good outcomes or meaningful outcomes, included more research on non-speaking or minimally verbal populations, and created new measures because oftentimes tests and tools are lacking to study minimally verbal populations. We also have started a study in low and middle income countries, in particular in South Africa, to look at the role of environmental factors in social, emotional, cognitive and behavioral development, and also started to run a clinical trial. And the main goal was to see whether academics can run a randomized control trial to industry standards. I just spent the last few minutes talking a little bit about the experience with autism representatives. We started off with 41 autism representatives that were included autistic people, family members, and autistic researchers. And it was, to be fair, a slow start. There were a number of concerns from the AREPs, some of which they've now shared with us, including concern about tokenism, a tick box being only involved at late stages, general concerns about being involved in a biomedical project. There was sometimes a tense atmosphere. There was about 2 years ago, so also, of course, Spectrum 10K and concerns about that. But also, it was really that at times we were speaking different languages. And I just wanted to say that it was, not surprisingly, a personal experience that really had tipped it off for me in terms of my stance on the language used. And this is, during that time, I also was in the process of adopting a child. I started to read more through the adoption literature and got involved with that. And I became aware that quite often the term broken or damaged was used for adopted children or children in foster care. And that was particularly the case for children who were over 2 years of age, who were a boy, and if they were in multiple foster placements and non-white, they were kind of like supposed to be on the damaged route. When I was then saying that I was adopted myself, then people would say, well, you are an exception, which is only of some comfort and in a way kind of helps to perpetuate stereotypes. And then I was able to find my son, and he's beautiful, and he was 6 when he first came. And I'm so proud of him, and he's doing incredibly well, and he's most of the time happy and making other people happy. But more to the point is I realized how the description of being damaged or broken is in itself so damaging, and how I would want to protect him from having to deal with that kind of impact on one's self-esteem, alongside with resilience to other early experiences. And I guess it was really the turning point for me when I really felt also incredibly sorry for having used language that, autistic people find as harmful and as hurtful, and how difficult it is to look beyond the language to understand what the person is intending to say. So since then, back to the work with our autism representatives, we have now got, I think, 8 or 9 working groups, including all sorts of different topics, from ethics and data sharing, research break times, biomarkers, autistic burnout, a clinical trials advisory board, policy making, outcome measures, and intellectual disability. It's probably fair to say that some of the working groups are more successful than others. So, for example, the intellectual disability group, led by Jenny Cook, is very well received, and others are less successful. I've been particularly interested in biomarkers, and we've started recently a community project to explore community views on the scientific, practical, and ethical questions of different biomarkers for different intended applications. This project is still in the pilot phase, and it was designed together with some of the AREPs who had originally participated in the interview that I had created, but gave a lot of very helpful feedback on how the interview can be improved. And this is now how we are going into the next phase of the project, where we're really going systematically to these different types of biomarkers used for different applications and see what are the acceptability and the concerns for these different scenarios. So as an interim reflection, so both the experience working with autistic people and also some of our findings had made me think about, where are we in terms of our biomarker approaches? And one possibility is that in the whole of neuroscience, reductionism is probably a predominant paradigm, which tries to understand complex systems by dividing them into the more constituent units and their interactions. But I began to wonder whether the focus on specific parts of an individual, whether this is like a particular cognitive process or a particular brain structure or brain network even, might help us or might lead us to neglect the interactions between the different parts that might produce a whole that is bigger than the sum of its parts. So what we call sometimes emergent properties. And that's particular to do with, for example, the dynamic interactions between the brain and the body. And then second of all, the context or condition in which particular characteristics operate also need to be taken into consideration. Oftentimes, we've been thinking of biomarkers as a more static snapshot at a particular moment in time, representative for this person's developmental stage, but perhaps we might have to look at it more in terms of a biological response to a particular environmental situation. So, for example, how brain and cognitive development are affected by acute stress and persistent stress. And then finally, also, I guess we had looked at the autistic person and including their social development, oftentimes in social isolation, which is in a way a little bit ironical. By doing so, we may have neglected to look at the interaction between the person and the environment. So before I conclude, I just wanted to give you another example of a great study by one of our early career researchers, Teresa Del Bianco. And so often we're asking loads of things about our autistic participants. And very recently, she designed a questionnaire that she gave to our researchers in our network. And 124 researchers took part, and she was interested in better understanding who we are. And the results were quite, in a way, probably quite surprising. So she found that 86% of the researchers said that they had at some point in their life a formal diagnosis of a mental health condition. Then 23%, so almost a quarter, responded that they either have a formal diagnosis of autism or identify as autistic. And up to 80% said that they had a family member that was either autistic or had another neurodevelopmental condition in their family. So this suggests that the distinction that we sometimes have made between researchers versus AREPs is not so accurate, because AREPs could have been researchers as well, but researchers also could be autistic or neurodivergent, but also that the distinction between autistic people and parents is sometimes unhelpful and inaccurate, because oftentimes people wear both hats. So then just to come to the conclusion. So then, so really what I think I'm enthusiastic to move forward to is to try and integrate a neurodiversity affirmative approach with precision support, which I think at the intersection recognizes differences with varying, recognizes differences, and that they can involve different degrees of support needs. And there is a common emphasis on tailored support. So the conceptual framework would integrate both social and biological aspects, because I think neither of them alone can be the complete pictures. I think it's important to bear in mind that biological models or biomedical models, even though they have been the same as have been done within a deficit framework in the past, do not need to be the same. And we can look at mechanisms and treatments for some autistic people and some features where they are desired, at the same time as advocating for a reduction of stigma. And also that the concept of neurodiversity is inherently inclusive, including the whole of the community. So how we conduct research is to really work from the outset with neurodivergent people to increase the face validity of concepts and of our methods. So we need to develop new measures that allow us to more frequently sample processes in different contexts, and new models and methods to study interactions. And then really to the end, one of the examples is where we're trying to do that is in a new network called Respect for Neurodevelopment, which was funded by the EPSRC and the MRC as a UKRI network, and that tries to bring together, in this case, bioengineers, psychologists, psychiatrists, and related disciplines, as well as neurodivergent families, to co-develop responsible, reliable, scalable, and personalized new technologies for neurodivergent children. So then in summary, I think my research was influenced both by general concepts, trends, as well as personal experiences. I think that neurodiversity enriches research and has prompted us to review assumptions. I enjoy working with autistic collaborators. And I think a neurodiversity affirmative approach to precision health makes me feel as a researcher more whole and happy, as well as a person.
Okay. Thank you very much, Dr. Eva. So here come the parts for the Q&A. Well, we have a question from Dr. Jyoti. She wanted to be part of innovation group for the medical part of autism research. So how can she be part of this research?
Sorry, I've missed the first part because I tried to look into the chat. So somebody wanted to participate in
your innovation group for the medical part of autism research. She herself is eating disorder consultant. So how can she be part of, how she can reach out?
For Respect or for Aims to Trust, but in either case, she's very welcome to send me an email.
All right, thank you. And another question is from Gabby. Is it safe to look for autism biomarker when so many groups would like to eradicate autism? Would it perhaps be better to work on wider social acceptance first?
So how much time do I have to answer this question?
Something very brief.
Okay, so what I try to say is that a biomarker is not a biomarker. You can use, for example, a cognitive test in order to make a prognosis of what kind of school support is most useful for a particular child. But we are talking here probably specifically about genetic markers, and we're talking about prenatal testing or using genetics either prenatally or postnatally. And I think this is a big issue, and that is something that we're trying to address to some extent with this community project about, desirability, acceptance, and concerns about different biomarker use. When we say that we don't think it's likely that there is going to be a genetic marker for what we currently call idiopathic autism, that's really only part of the story. I think it really requires a fundamental comprehensive discussion between researchers, the autistic community, ethicists, and everybody that is involved. In that, I do believe as well that the perception of what is autism really would impact if a prenatal marker was available, whether or not it was desirable. So, for example, there are already prenatal markers that can tell us whether there's a boy or a girl, and there are some societies where this might lead to more specific terminations, and in other societies it's not. So I think it's both scientific advances and a discussion alongside with social implications that have to go together. I feel I cannot really answer this really important question very well in a short time. Maybe when we have the panel in the afternoon.
Okay, all right. So you mentioned that following the adoption of your son, you regretted some of your language used in your autism research. Do you also regret having been involved in some of the autism research projects you have contributed or on contributing to? This is something personal.
No.
Right on.
Yeah, no, I don't regret that. No.
Okay. All right. Last question is, what are some major positive factors in increasing emotional resilience in autistic people? As in, what helps autistic no breakdown as easily, and what helps ease bounce back?
I'm reading the question again. What are some of them? Yeah, so I mean, resilience is a really important issue, and I think it's both environmental factors and, in fact, factors in a person that could help increasing resilience. So, for example, we're talking a lot about sensory processing, predictability. So if there is an environment, for example, where sensory overload can be minimized because people are aware of the impact on that, on functioning. So, for example, things like concentration, attention, memory, one can function very well in one situation, and the whole thing can break down in another situation in the same person. So in order to minimize some of the factors that can lead to impact on other cognitive functions could really help to increase resilience.
Thank you, Dr. Eva, for your time and giving us this much insight about the research happening. And we are going towards the lunch break, and we'll be back at 1:15 p.m.